Hi all, this is Group 21's Vaccine RR's team proposal video. The WHO has collected data stating that in 2020, 10 million people fell ill with tuberculosis worldwide. A total of 1.5 million people died, with the highest rates of infection being in Southeast Asia and areas of Africa. These figures need to be lowered. The aim of this challenge is to produce vaccines to decrease infections and deaths, since antibacterial tuberculosis resistance is increasing. Furthermore, the BCG vaccine, a widely used vaccine containing a weakened bacteria, is unreliable, so the world needs a booster. But where would we produce these vaccines? In Uganda, a vaccine plant will increase vaccination rates and produce jobs, increasing economic development and national infrastructure. The challenge is split into three essential parts: heating, identifying how heat is transferred through the bioreactor; mixing, investigating different reactor variables such as propeller design and speed; and the probe, experimenting with yeast cells to investigate cell growth through impedance. How many people will we protect? The whole population of Uganda. The vaccines will be safe for everyone. Protein-based vaccines are safe for children, and with herd immunity, the more people vaccinated, the more effective it is. Furthermore, regarding the bioreactor facility, ImClone was chosen as it fitted the liter requirements and had the lowest batch number, which is essential when accounting for faulty batches.
Why do we need the probe? It is required to monitor the growth of its cells, since our aim is to generate a maximum amount of vaccine via these genetically modified yeast cells. To ensure the most efficient production of vaccine, the concentration of yeast has to remain high. Therefore, we would need to dynamically monitor the growth of these yeast cells to prevent killing them. How can we monitor it? We use the electrical impedance. Body tissue can be simulated and represented by this circuit, and we can monitor the impedance across it by applying Ohm's law and assuming the current is fixed. The voltage across the tissue is directly proportional to the impedance, so we can monitor the impedance by measuring the voltage. Then how can we understand cell growth from impedance? We can apply this formula for the resistivity. At low frequency, when the area or the extracellular space decreases due to the increasing number of cells, the resistance increases, making it harder for the current to pass, helping us monitor the growth. At high frequency, the current will directly pass through the cell membrane, dropping the impedance. Hence why we need to find out the optimum frequency for a clear signal to be observed. It is tested using my deck. Then we start measuring at the first detectable frequency and its corresponding voltage. After that, we stepped up with interval of 500 Hz. The increase in frequency caused the impedance to drop, which drops the voltage. As the voltage starts plummeting, we record more readings at smaller intervals, then proceed to plot an output voltage against frequency graphs, finding the most suitable input voltage and the gain to observe a clear signal. That we have to experiment with different input amplitude and gain. Next, the electrode placement. And put the sensor electrodes near these two electrodes, so it is sensitive enough to detect, but not too close because we might measure the impedance of the electrodes. After these factors are fixed, we start doing a calibration curve, use different concentration of each solution. These solutions will have go through the test, and then we plot the output voltage against concentration curve with the data collected.
Heatsink subtask. Yeast growth is optimum between 30 to 35 degrees Celsius. Any higher will cause the rate to decrease and eventually kill the yeast. Any lower would slow the rate of reaction. Our target temperature on the hot plate is 35 degrees Celsius because heat loss would reduce the temperature to 32 or 33 degrees Celsius. The hot plate also allows us to put either 30 or 35 due to its calibration. As shown, PET plastic has a lower thermal capacity than cardboard, so it will be used as the lid. And for the plastic cup insulation, out of our materials provided, we will test various forms of insulation that is shown using 35 degrees as our temperature. And insulating the bottom of the cup would just increase the time to heat the cup. Next, experiment planning. After finding the best mixing and insulation method, we will conduct these 4 experiments. For this, we will need to assemble the apparatus, then add the yeast mixture and measure the initial temperature and the top and bottom of the yeast mixture. Then we switch on the hot plate and measure the temperature of the top and bottom of the yeast mixture at every 2-minute interval till we reach 10 minutes. Fourier's law is used to interpret the results since it shows the proportionality between the thermal gradient and the heat flux. We then create this table accordingly for all our 4 experiments. The values -K, A, and dx should be constant with these values. Hence, we plot the graph of heat flux against dt, where dt is the difference in top temperature from the bottom. Lastly, anticipated results. The gradient will be -Ka over dx, and we should aim for a negative linear graph, where the steeper is better because it indicates that the top and bottom temperatures are the same.
Mixing is important as it allows us to create a homogeneous environment for the solution, which ensures consistency in all samples taken from the reactor. Mixing allows aeration, which helps with gas blending. It's important that we test for the optimum conditions of the bioreactor. To test for the most optimal impeller, we will control the agitation speed to 200 rpm and then perform dye dispersion tests to find the impeller which gives us the shortest mixing time. We will then repeat the tests under baffled conditions. Multiple repeats of each test will be done. 2 of the most optimum impeller will be used and a baffle if it is effective. Liquid level is set to 8.4 centimeters for optimal yield and mixing time. A vortex prevents uniform mixing. Baffles should help prevent the formation of a vortex at higher agitation speeds, which allows uniform mixing and short mixing times. Graphs can help us interpret results. A bar graph with standard deviation bars can show if there is any significant difference between baffled and non-baffled conditions. A line graph can help us to compare different agitation speeds, which will allow an optimum to be found. Too high may kill yeast cells, too low gives long mixing times.
Lastly, conclusion. Both heating and mixing are connected to the probe via the growth of yeast cells, whereas heating and mixing are connected to each other via the heating graph.